Our next talk is given to us by Lydia Bright, and it is the functional diversification of Rab GTPase paralogs in Paramecium.
Okay. Hi, everyone. So is it up or down arrows or left or right? Okay. Awesome. Awesome. Okay. So I think Marcella showed this earlier, but it's such a fun image. So unicellular eukaryotes are both diverse and highly complex. And shown here is an atlas, a visual atlas of just some ciliate cells drawn to scale. And you can see here that ciliates are both diverse and morphologically complex. And what I'm really interested in general, is how evolutionary forces interact with subcellular and cellular constraints to drive changes in function that lead to these changes between cells and the complexity that arises in cells. And specifically, I'm interested in how evolutionary forces are acting on the Rab GTPase family in Paramecium species, and also whether they've changed in function from their duplicates. So a lot of this functional change occurs as duplication and diversification of function happen in gene families. And that's what I'm focusing on. So Paramecia, as you probably all know, are large swimming bacterivores. And I know Ron said that ciliates are sensory cells. I think ciliates are basically phagocytes. I think of them as eaters. They are very efficient at taking up and eating bacteria. And so here, you can see the phagocytic pathway better. So here's some food vacuoles, and here's a forming food vacuole at the base of the oral apparatus. And also importantly for later in my talk, Paramecium cells as freshwater-dwelling protists have two contractile vacuoles that control the osmolarity of their cytoplasm. So they pump water continually out of the cell. So the Paramecium Aurelia species complex is a really good model for my purposes. It emerged following 2 whole genome duplications. And there are 15 species within the complex. And from the last 2 whole genome duplications, about 50% of the duplicates have been retained. And to me, this equals a lot of parallel evolution to study at the subcellular level. And I've chosen to focus on 3 of the Aurelia species for the purposes of this study. Let's see, Tetraurelia, Biaurelia, which are spread across the phylogeny, and Sexaurelia. So on an individual level, a gene duplicate can have one of several fates. It can be lost or non-functionalized through non-functionalizing mutations or pseudogenization, or it can be preserved. And if a duplicate gene is preserved over time, it can be preserved with no change in function or expression level or pattern, or with a change in function, which can take the form of neofunctionalization or subfunctionalization. And then here's a little bit of background on membrane trafficking, although Sabrice gave you a little jump in, which is great. I don't usually get that. So each step in eukaryotic membrane trafficking, so from each individual compartment to another, consists of a budding or coat-forming step, and then transport across the cell, and then fusion with a downstream or acceptor membrane. And I'm particularly interested in Rab GTPases, which are the proteins that determine where vesicles will fuse and what downstream membranes they will fuse with. So they confer a lot of the specificity in membrane trafficking in the cell. And so as a result, individual Rabs, so shown here as red numbers in a prototypical eukaryotic cell, control only one or a small number of trafficking steps. And for my purposes, this is really useful because this means that a given Rab's gene family member's subcellular localization pattern corresponds really tightly to its function. So when a Rab or a Rab duplicate or paralog changes function, its subcellular localization may also change, will probably also change. So to get at my first specific question, how are evolutionary forces acting on the Rab GTPase family. I'm just going to give you a summary of some work that Jeff and I have done because I don't have time to go into it today. But we found, so this is work with Jeff, we found that the Rabs in the 3 species that I talked about, Tetrarelia, Biaurelia, and Sexarelia, are on the whole, each Rab gene family is much more highly expressed than other genes in the genome. They're also more highly retained from the whole genome duplications. And also, interestingly, their expression levels between the duplicates are less correlated. So they're more divergent in expression levels from one another. So these 3 things together are consistent with a model in which the duplicates are retained long enough to acquire expression level and/or expression pattern or other functional changes over time. So this may set them up for further functional change. So to get to my next specific question, whether specific Rab GTPases have changed in function from their duplicates, I'm going to get into my localization studies. And for this, since I can't look at every one of the 170 Rabs in each Aurelia genome, I chose to focus on the Rab 11 subfamily. And if you look at this chart is from Tetrarelia, and it's the number of Rabs in each subfamily. And you can see that some subfamilies, so Paramecium are in red, Tetrahymena are in orange, a vertebrate is in blue, yeast purple, and plants in green. And you can see that subfamilies contract and expand over eukaryotic evolution. And in particular, there are 5 subfamilies that have expanded in Paramecium, the Paramecium lineage. And some of these expansions are shared with Tetrahymena too. And then also some are parallel expansions with plants or animals. So I'm focusing on the Rab 11s. So Rab 11s are really important and central, and Sabrice already mentioned them. They direct traffic to and from recycling endosomes. So they direct traffic, a lot of receptor recycling back to the plasma membrane, and other traffic recycling back to the plasma membrane in all eukaryotic cells. They direct traffic to and from sorting endosomes, for which there's not an arrow here. They also direct traffic from recycling endosomes back to the trans-Golgi network. So they have a really central role in these compartments in this part of the endocytic pathway. And as a result, in different eukaryotic lineages, Rab 11s have taken on different functions as well as their conserved functions. So different Rab 11s have diversified. And they have roles in long-term potentiation, in pathogen resistance, and phagocytosis in other lineages. So to me, this was a really good candidate subfamily to look for this diversification. And in terms of the genes that I chose to focus on, so they're in green on this gene tree. So these are representing genes from Sexarelia, Tetrarelia, Biaurelia, as well as the 2 outgroups that were on that earlier tree, Caudatum and Multimec. And remember that these predate the 2 whole genome duplications, which I have here. So the 7 genes that I chose to localize for this study are in green. And I made GFP fusions of each one and looked at them in their native cells. So each one is localized in either Biaurelia, Tet, or Sex, depending on the gene. And actually, Sabrice, again, gave a really good introduction to lysotracker, which is this red acidic marker dye. So just to orient you to the cells. So when you were looking at the cells, I try to make them all in the same orientation, but it's hard. Here's the macronucleus, and here are some acidified food vacuoles, which, as you probably know, don't stay acidified forever, so there are also food vacuoles that are not acidified. And the lysotracker also labels lysosomes, which are the smaller vesicles, and it also labels other parts of the endosomal pathway, including sorting endosomes. And also I've put arrows next to the 2 contractile vacuoles in the cell. And here's the base of the oral apparatus and the oral apparatus. And so here's one of my fusions, the Biaurelia 11A. So it's from this, let me go back, actually. So these are the 3 clades that I looked at of the genes, I, A, and C. And you can see how they're related. So these are the most closely related genes within this subfamily. And you can already see that there has been loss, and some of the clades are bigger than others. So here's one, and here's the lyso tracker. And then when we overlay them, we can see where the overlap occurs and make decisions about what compartments they're going to. So in this case, based on morphology, we can see that this fusion goes to the base of the oral apparatus as well as filaments extending from the base. And when we do time lapses, which I'm not going to show you today, sorry, we see that the green is going back toward the base of the oral apparatus or recycling back. And we would guess that it's involved in recycling of vacuolar membrane back to the base so that those big food vacuoles can continually form from the base of the oral apparatus. We can also see that it localizes to, so it's co-localizing with the lysotracker at these big tubulovesicular complexes, which I'm guessing are sorting endosomes. So not co-localizing with food vacuoles that are acidified, possibly co-localizing with some lysosomes. So when we look at all of the orthologs and paralogs from the eye clade, we find that they all share a nearly identical localization pattern. And that's the one that I just described to you, this the base of the oral apparatus, filaments extending back to the base, and then the sorting endosome co-localization with the lysotracker. So they're all, it's a pretty broad pattern, but it's very specific to these compartments. So when we look at here's an example of 3 out of the 4. And hopefully you can see that they're pretty similar to each other. But when we start to look at the next clade, from which we've just localized one member so far, so a representative member of these 3, we see that it actually has an identical localization pattern to the other clade, this broad pattern that involves both sorting endosomes and the phagocytic pathway. So at this point, since I'm looking for functional diversification, you might be wondering if I ever found it. And here's the comparing to the other clade. But when we did look at the third clade, we found that there was a different localization pattern in these 2 proteins. So it looks like this one, these members are going to tubules emerging from the contractile vacuoles. It also has some cytoplasmic localization, which is why I'm not showing you the lysotracker here, because it would look yellow everywhere. And then also possibly to the nuclear membrane. There's also some localization to the base of the oral apparatus still. But there is some functional diversification that has gone on or is going on here. So in conclusion, while members of 2 out of 3 of these larger clades had virtually identical localization patterns, one had a different but still specific pattern. And so this indicates that this clade has functionally diversified or is, and we're going to be looking at other members of this clade to see if they all have the same pattern. Also, it's worth noting that we haven't found diversification between the products of the whole genome duplication yet, but we're still looking. We have a lot of tagged Rabs to go through. So. Oh, and also, the relatedness of these clades now allows us to make inferences about the ancestral state of these proteins, right? So since these 2 clades are more closely related, and these 2 share this conserved localization pattern, we can infer that this was the ancestral pattern and this is the clade that has diversified. And we can also look at the outgroup species to see if that holds up. And also, more future work, these functional changes appear to correlate with a small number, actually 3 amino acid changes in key protein binding domains of these proteins. So we can start to change these residues and see if we see these localization changes if we just change these 3 residues. So that's our next step. And the large number of compartments labeled by those conserved protein localizations can tell us a lot about the close relatedness of these endocytic recycling and phagocytic pathways. So this is one of the benefits of this kind of work. We can learn about the ancestral state of these compartments, start to learn about the function by comparing proteins localization in different cells. And I'd like to thank people in my lab. You can tell that I wouldn't have been able to do this without Jeff, Tom, Parul. Ugna did a lot of the cloning. Now she's a tech in the lab. and undergrads that did the cloning, and also a shout out to my undergrads that are here. Catherine and Rainey are going to give talks on Sunday, and Athena is giving a poster, and also funding sources. And lastly, just like Jeff did, I'm going to give a plug for a job opportunity in the Lynch lab. So I'm moving on to a faculty position, and my position in the Lynch lab is open. Thanks. Wait a couple years, then you can clap. We'll see. But the Lynch lab is a great dynamic place to work. Always people to talk to, and we need a molecular biologist. So we've got lots of evolution going on. But I'll take some questions. I have 25 seconds.
A quick one. Are there any good markers for some of the endosome compartments that you're going to be bumping into, like this sorting endosome compartment?
That's a good question. There are, I think, there are the general ones, which is like the mitotracker or the lysotracker, and then when you get into the more specific, you're going across a lot of evolutionary time. So, some of them are specific to certain proteins in other cells. So, yeah, it's something I'm looking into. I don't know for sure yet.
All right. Thank you very much, Lydia.
Thanks.
Okay.